want to move forward with what we're noticing in disease-modifying therapies and what's coming down the pipeline. And the first thing that comes to mind, and there have been questions in the Q&A about this, is cell replacement. Cell replacement treatments are one type of disease-modifying treatments because we know that in PD, there's a loss of dopamine-producing cells in the brain. So in general, how are cell replacement treatments supposed to work? What can we expect from this type of treatment? What are the risks, complications?
Yeah, and something that's getting hot again, right? And so we were cold for a while, right? Meaning that in the past several decades, stem cells and circuit-based therapies, a lot of people believe they were going to be the answer. A lot of fundraising happened in that area. A lot of folks, put their hearts and souls into thinking this was going to be the next gen, and it really didn't happen. And so now when you think about the rebirth, and we talked about this in our new book too, kind of the rebirth, a lot of the R's, but rebirth of these, of thinking about these, types of approaches, how they can be useful. We have seen the emergence of several approaches that are now reaching or coming close to, and in one case into the market. So I think it is worthwhile to just take a moment, take a breath and say, okay, what's going on? So what's going on is you take a stem cell, which is like that early cell, you can turn into whatever you want to turn it into, okay? And you can use it to replace dopamine in the brain, okay? Mm-hmm. And so the idea of at least the ones that are out there on the market, and there's actually some other ideas too, but the ones that are getting hot that people are now thinking about, they're going to be the R. They're going to be replenishing lost dopamine producing neurons. Remember, Parkinson isn't just a disease of dopamine, but it is a strong, part of the circuitry and could be very important. And so the goal is to restore what's called endogenous production, what should be going on in your brain, okay? And so there have been early trials now. And so there's a couple of places, people have heard this term, Blue Rock, Aspen, the Japanese trials, and it gets like confusing because if you're a person with an individual with Parkinson, or you're thinking about one of these things, what are they? Okay. And so let's kind of walk through a bit of those, because I think it's probably worthwhile just to take a moment and take a deep breath. Blue Rock is a company that has a drug. It's called Bananaprosol. I don't know if I'm saying it right. Of course, all these have names. So you and I both struggle with this, every year, and that never gets any easier during our 5 years. But BEMDANEPROCEL. Okay, so if you read that or you read Blue Rock, which one are you talking about? Well, in this case, this is what's called a donor-derived cell, Sonia. So this cell comes from an embryo, right? It's an embryonic stem cell. And then it's going to be transplanted into a part of the brain called the putamen, okay? And the idea is it's going to do the R, another R. It's going to try to rebuild the circuit. Phase 1 is safety, and it showed good safety and tolerability. It's they've done some imaging to show that it can survive. There's been some early signals of improvement, even out to 2 years in this. And one of the things that's important when you talk about any of these is whether or not they need immunosuppression. And this particular one does need immunosuppression. So that's like if you got a heart transplant or a liver transplant, your body may reject those, the tissue or the cells. And so in these cases, they're talking about it typically a year or so of immunosuppression, where your immune system needs to be suppressed. And now it's in a phase 3, which is sort of that trial before they're going to be looking to see, could this be for dopamine? Remember, it's not just a disease of dopamine, but for dopamine replacement, could this be a potential path forward? There's another, one called Aspen. Okay, so like our course, so we have the Aspen course, but this is called Aspen Neuroscience, and they have what's called an autologous type of approach to a stem cell. Okay, an autologous means it comes from you yourself. They make it out of yourself and they put it back into you. Okay. And so when you're reading about this, many people will come into our clinics and they'll say, hey, Dr. Okun, tell me about ANPD001. And I'm like, which one is this? Okay. So that, if you read that, that's the Aspen one. That's the one that comes from the cells itself. It has an early name called Bananaprosol. Okay, I'm not sure I'm saying that right, but they both end in PROCEL, okay, cell replacement, right? And it's coming from your own cells. They're going to re-engineering them, another R, Okay, we use the R's a lot in our book when we talk about, these types of therapies in the Parkinson plan, but we're going to reimplant them into the brain, rebuild the dopamine circuitry. And because it's your own cells, you don't need immunosuppression. Okay. There's 12-month data on these, and it seems to add a couple of hours of good on time, per day. And the imaging, just like in the other trial, tends to show that these grafts are surviving and they're moving toward phase 3. And there's currently, I think, only about eight patients or so. There may be a few more now that are published. There were a couple of papers about these approaches in Nature, medicine. So it's sort of coming back around. And then the one that hit the news most recently, Sonia, was Japanese.
Right.
And coming from the Kyoto University. And of course, we should remember that from that university, a very famous Nobel laureate was the one that really, brought us into the stem cell era, right? And so there's a long history there in Japan of great science on this. And so they actually have something for stem cells, and these are stem cells that are derived from donor cells. Okay, they're not your own cell. Okay. And if you read about it, they usually call it in the papers or the literature, or if you're reading about it, they'll either call it the Japanese approach, the Kyoto approach, or they'll call it AMCHEPRY, AMCHEPRY. Okay, so if you see those, you know that so now you see why it gets confusing. So, all of these names and everything, right? They're implanted into the brain. They're going to do an R. They're going to restore dopamine production and neural networks. And what is different is, based on the early trial of 7 patients in Japan, they have a regulatory system where they can actually get some pre-approval to allow it to go in and allow people to get it and for them to collect data on it. And so they will do that for 7 years. So with 7 patients, now they're going to do this for the next 7 years. So we're going to get data longer term as it comes out, and we'll see what happens. And those early patients, 4 of 7 patients, had some real robust biological effects. And again, this is not autologous, so you might need immunosuppression. So it's something to watch, whether you're going to need to be on any of the medications. And the approval here is conditional and time limited, depending on real world data. And so we'll see how these things, do. And so thinking about it, those are the 3, and the 3 that, how I would end. I'm taking a moment here to explain that because I think it's important. Last point. Parkinson is more than a dopaminergic, issue. So just replacing the dopamine doesn't cure the Parkinson disease, can have powerful symptomatic benefits. Here are 3 stem cell therapies that are moving closer to that benefit. Will it be for you? Will it be better than DBS? Will it be better than these other things? That remains to be seen, and it should be a discussion with your teams if you decide to move forward for these things.
And do we have any long-term data then at all with stem cell replacement?
We have some, and of course the early long-term data is not as good because they hadn't refined the process. Many people were getting something called runaway dyskinesias from some of the early trials. There were 2 really large early trials from this. And so this is sort of the next gen of these therapies, and so thinking about this, but as you mentioned before, it isn't a cure. It's more than a dopamine disease. And so a lot of times the hype doesn't live up. And so we want to just make sure everybody understands what we're talking about.
Right. And another just quick question about that. You mentioned that like the Blue Rock was using embryonic stem cells, and the Aspen was using autologous, and then Japanese was using donor cells. Is there an advantage to any one of those approaches that we know of? I mean, you don't have to take the rejection drugs potentially.
Yeah, so each group is going to argue their point. It's kind of like go to court of law and say, oh, this is my point this way. So they're each going to, argue their different points. The main one is the immunosuppression. There's some attractiveness about, taking your own cells and putting it back into you. So I think that has some sex appeal, but whether that will be better or worse or whatever, we don't know. I mean, there are so many things in medicine, and you and I are both doctors, and that, like seem logical, but don't end up being, what we thought they were going to be. And so I do think we have to wait for the data, on the comparisons. What's nice is, how much now with the internet and with sharing, and you and I grew up in medical school before there was PowerPoint and a lot of emails and everything. Now we can share data, so we can see it really quickly. So if you want to wait a little while and watch what happens to a bunch of these folks in Japan, and you want to wait for some of these trial results, wait a little bit. We're going to start seeing some results, and use that data to your advantage, and enroll in the clinical trials, become part of the solution.
The other thing that in relation to the stem cells is that this is a question from one of our audience members, that the Aspen study is also for patients who have been diagnosed for over 4 years. Why is 4 years diagnosed so important, and are there other options for folks that are diagnosed in the last 4 years to join studies? Are there other studies?
Yeah, so it's a great question. It turns out that how you assemble a trial and who you include has so many pieces that will impact whether this trial is positive or not, because you have to define a population, and two, with the regulatory agency. So it could be that a lot of the folks that are getting into this, the regulatory agencies, are going to have you choose a population where you're going to cause the least amount of harm. So it might not be the population who needs the therapy the most, but you start with the people that you're going to cause the least amount of harm, have the lowest risk, to be able to do this. One of the things that's frustrated our scientists in the laboratory here at the University of Florida and across the globe has been, this idea of early, right? If you want to get something, you go in as early as you can. But when we think about regulatory agencies, there's some, hesitation to going in too early. We've also had real tragedies where we thought people had certain diseases and they didn't. And so many of the Parkinson drug trials, don't want to shock anybody from, the past couple of decades, as many as 15 to 20% of the people enrolled may not have had pure Parkinson disease.
Right.
The 4 or 5 year, window isn't always used in this way, but I'm going to tell you the most common reason that people end up using a 4 or 5 year window. It's because if you wait 4 to 5 years, statistically, those people will end up having Parkinson's and not something else, a higher statistical probability. And early on, when surgical therapies came out, like DBS, they would make everybody wait that 5 years. Turns out that people can even convert to other things after the 5 years, but statistically, you get a better group. As we have biologics, the ability to do, skin biopsies, blood, CSF, other things, this may help us, and genetics, to be able to better define these different groups. But those are some of the reasons. It's a wonderful question.
Yeah, the biomarkers will really help us delineate those groups without having to wait the 4 or 5 years, which I'm looking forward to. Then when we talk about cell therapy, there's also gene therapy. And I would want to ask you if you could discuss how this differs from stem cell therapy. I mean, genes, I know, carry our blueprint, determine everything about us, our hair color, our eye color, our potential risk for developing a disease. But what is gene therapy, and how does it work in the face of Parkinson's?
Yeah, so nice, easy question for me, Sonia. Like right down the middle of the plate, right? Gene therapy. Let's dig in a little bit. And so basically, you want to get something to the brain, okay? And so before you're just giving a cell, you're going to replace a cell, you're going to try to regrow it. In gene therapy, you're going to take something called a vector. A vector is something that is going to deliver. It's like a delivery truck. It's like a UPS truck. You're going to package something on that UPS truck, and you're going to send it into the brain, okay? You can deliver genes, you can modify neurons, you can try to enhance the way that dopamine is synthesized. You can alter these pathways, these networks that we've talked about that are so important to being online in Parkinson. And when they go offline, you get different symptoms. And in general, you're hoping when you use a gene therapy, you're going to get some long-term modification of the brain circuitry. Now, the thing that's really tricky about gene therapy is sometimes it can be irreversible. The delivery, getting it to the place that you want, and do we know where that place is, and the mechanisms. And there are a lot of scientists who are really hard at work now, and how you get things to the brain is actually harder than you think, and so pushing it in and just waiting has been a real tricky thing. So there are some big picture points that I always like to make in talking about gene therapies. We should think about them into three strategic buckets. You and I both like buckets, right? And so bucket number one is enhanced dopamine production. Okay. And so if you read about gene therapies like AADC, amino acid decarboxylase, or prosavin therapy, they're going to boost your dopamine production, okay, and replace dopamine synthesis. You're looking for a symptomatic benefit here. It's likely not disease modifying in that case. If it is, it's very mildly disease modifying. The second picture for gene therapy is circuit reprogramming, okay? And so those are the therapies you're thinking about, like glutamic acid decarboxylase or GAD, where you send in a gene and you actually reprogram it. So before it was an excitatory chemical and you change it to an inhibitory chemical. And so this was done in a very famous experiment in Parkinson's disease with GAD therapy into humans, and by Michael Caplet and colleagues. And the idea here in circuit reprogramming is you're going to try to normalize the abnormal structures, those abnormal circuits that we refer to as the basal ganglia. Not important that you know that, but these abnormal circuits. And the strength of it is it's kind of like DBS without hardware, okay? And so we think about that. The limitation is it's not going to address the degeneration, okay? The third bucket is neuroprotection or disease modification. The bucket that Soania, loves the most, right, is the GDNF, the glial-derived neurotrophic factor, GBA1. Okay, you remember the GBA1 mutation, right, coming back around here. The idea is that you're going to use the gene therapy to slow or reverse neuronal loss, and then you're going to try to get to some true disease-modifying potential. And one of the biggest limitations, Soania, on this is it's really hard biologically for us to prove that we accomplished what we said we were going to accomplish. Now, if you are a person in the field that is reading, or you're an individual with Parkinson, you may see a number of these things come up, and it's confusing because there are so many of them. Okay. So if you see something that has the word AAV, Soania, on it, it's usually a gene therapy. Okay. So if you see something that says AAV2 version 2 dash AADC, that's usually the amino acid decarboxylase gene therapy. Okay, putting the enzyme into the brain. If you see something like AB, like the alphabet AB1005, that's a GDNF therapy, and it's just hard for people to know. Like when you're reading all these things, that's a vector that's using a virus. Okay. An adeno-associated virus, like adenoviruses can cause colds and things like that, to promote neuron survival by giving GDNF. And there is a trial called the Regenerate PD trial, and that is a gene therapy trial. And the idea is, again, to try to change that circuitry. If you see one that says AAV GAD, Soania, if you see that, AAV is adeno-associated virus, so now we know it's a gene therapy. GAD, remember I taught you GAD, glutamic acid decarboxylase, that's adding the GAD gene into the subthalamic nucleus, and there was a very important study. That was the study done by Michael Caplet. And so now you're sort of catching my drift here. So if you see AAV2 GDNF, guess what? That's one of the early GDNF trials where they're sending things in. And then the one that Soania, I think, was keying on a little bit here earlier and talking about, neuroprotection and things, is there is a trial, and sometimes you'll see it referred to as PR001 or PR006, or the Prevail. And it's an Eli Lilly, sponsored trial, and it's all about the GBA1 gene therapy. So we know sort of what GBA1 does. We know about this thing called the lysosome that helps us to recycle things and to clear off bad proteins like alpha-synuclein. And we know that the GCA, the GCase, glucocerebroside, we know that enzyme activity, by boosting that, we might be able to change this. And there's actually a trial of Embroxol, which is a cough medicine, to do this. But there's also a gene therapy trial. And so if you see the Prevail trial, or the PR001 or PR006, you'll know that is a gene therapy. And then if you've got one breath left, Soania, there is another type of virus. It doesn't have to be an adeno-associated virus. There's a virus called a lentivirus, different virus. And that's the one that, when you read about it, it's called prosavin, is that therapy. And sometimes OXB102. I mean, I find these things mind-blowing to try to keep track of all of them. But again, this one goes after multiple dopamine synthesis genes. So it's kind of an interesting one. So it goes after the TH gene, tyrosine hydroxylase, the AADC gene we talked about. There's another one, the GCH1 gene. And the idea is that they're going to try to promote more continuous dopamine production in a part of the brain called the striatum. Not important for people to remember striatum, but replacing a dopamine synthesis pathway is important. They're thinking about, like, is there like a dopamine factory, and can you replace the machinery in multiple parts of that factory? We don't know how these are going to perform. Any of these cell-based therapy, stem cells versus DBS versus pumps versus medications, they're not for everyone. But I do want people to kind of be aware as they're reading. The numbers on the studies usually mean it hasn't yet been given a name, okay, or hasn't made it into trial. And then once they're given a name, sometimes you can decode the name, whether it's a gene therapy or stem cell therapy in the name, like what's in a name, and sometimes you can't. And so just starting to sort out your buckets can be tricky. And I think we need to help people with that, Sonia, because I think it's a really hard one.
Yeah, no, I agree. But where are we in terms of gene therapy in general, in terms of the pipeline? Like, how are we what phase trials are we in general? Is it something that we can expect to come to fruition sooner rather than later, sort of, or are we at the very early stages?
Yeah, I mean, I think there's been a shifting landscape here, and the landscape shifts when we develop new tools too, right? So we don't have some of the tools and some of the delivery things. So we're getting better at delivery. We're failing a lot, and I don't mean to shock anybody, but we need a lot of failures to get to a lot of successes. Okay.
Right.
And we failed a lot in gene therapy. That's okay. And we're improving. We're seeing how these things can get in. We're making sure we get our delivery systems right. We're coming back to some old players like GDNF. We're coming to some new players, right? And thinking about, targeting some new things and what's going to be the next gen. And so it is in progress. I think in general, what we're seeing is scientists are getting more successful in making something work that has a safe effect and some positive effect. And the question is how much positive effect? And I hate to keep saying this, but will the juice be worth the squeeze, right?
Right.
And then when we think about genes, we have to think about, what does the gene do, Sonia? So does it make too much? Does it not make enough, right? So what does it do? And could we be using, CRISPR gene editing technologies and things to move this along? Or could we use these technologies to learn what these various things are doing within the brains? And so I would say you're seeing good safety data on a number of these things. You're seeing, not the efficacy data yet that you would want to see versus some of the other medications and some of the other types of therapies like DBS and things like that, but getting better and people coming up with more and more creative approaches. So I think it's very promising.
That's good to know. And I like that about, failures leading to success, because I think to learn from why something failed gives us just valuable information. And so I like that.